Chapter, we're going to be taking a look at the origin of biological molecules as well as the origin of life itself. Explain the origin of biological molecules such as carbohydrates, lipids, proteins, and nucleic acids prior to the evolution of life itself. We must assume that these molecules were formed abiotically or totally independent of living systems. In the 1920s, two researchers, Alexander Oparin and J.B.S. Haldane, hypothesized that the conditions of the early Earth were indeed suitable for the abiotic synthesis of organic molecules. Today's atmosphere is often regarded as being an oxidizing atmosphere, for it contains a relatively high percentage of molecular oxygen. Oxygen, as we should know, is highly electronegative and would tend to strip electrons away from molecules. Therefore, such an atmosphere would not likely foster the formation of chemical bonds necessary to form complex compounds. In contrast, Oparin and Haldane suggested that the early atmosphere was a reducing atmosphere, which lacked any appreciable molecular oxygen. In such an environment, they suggested the chemical bonds necessary to create complex compounds and, more importantly, the building blocks of all life could potentially form. The activation energy required for these bonds to be established was likely supplied in the form of lightning strikes. The subsequent raw materials for complex compounds would ultimately begin to collect and concentrate in the early oceans. In the 1950s, two researchers from the University of Chicago, Stanley Miller and Harold Urey, designed an experiment in order to test the Oparin-Haldane hypothesis to in fact see whether or not conditions of the early Earth could have given rise to the first biological molecules abiotically. Here we can see the setup that Miller and Urey used in order to test the Oparin-Haldane hypothesis. Some of the very important elements of this model include a flask simulating the primordial sea, an atmosphere containing the gases that were thought to exist in the early Earth's atmosphere. When the water in this flask was heated, it began to evaporate and was directed up into this chamber containing the gases simulating the primordial atmosphere. Electrodes supplied sparks simulating lightning strikes to supply the necessary activation energy for many of these gaseous compounds to potentially react. A condenser then directed any of these compounds back into the primeval sea, whereby they could be collected and ultimately analyzed. After about a week or so, it was detected that these samples contained almost all of the building blocks necessary to create biological molecules: amino acids, nucleic acids, various simple sugars, as well as hydrocarbons. The question that we need to ask, however, is how did these abiotically derived molecules assemble to form the first living things? The first step in the formation of life would have to be the appearance of certain self-replicating molecules. Now, as we should expect, such molecules would be prone to error during reproduction whereupon we should expect a wide array of variants to appear, all in direct competition for one another for vital resources. This undoubtedly would have set the stage for natural selection. The question becomes, what exactly were the first self-replicating molecules capable of initiating natural selection? Likely candidates for this first self-replicating molecule include DNA, protein, and RNA. With regards to DNA, it is unlikely that it would have been the first self-replicating molecule to appear, simply because it requires enzymes for its replication, so it could not have existed prior to the existence of enzymes. With regards to protein, it is also unlikely that it was the first self-replicating molecule, because it requires DNA for its assembly. So therefore, it could not have existed prior to the existence of DNA. With regards to RNA, it is very likely that RNA was in fact the first self-replicating molecule, because unlike DNA or protein, it can direct its own assembly. The RNA world theory suggests that RNA molecules were indeed the first self-replicating molecules to appear on Earth. RNA, as the first self-replicating molecule, would have been prone to copying errors during the replication process, thereby giving rise to an array of variants, all in direct competition with one another for vital resources. In this case, free nucleotides necessary for replication. Since some molecules may have been better replicators than others, natural selection would have favored these molecules, and their frequency would have begun to increase. We also could imagine that some of the best replicators may have been able to actually degrade other rival molecules. Whereas some RNA molecules would have been susceptible to degradation, others would have been protected by being encapsulated in phospholipid membranes. Within these RNA survival machines, these molecules would have been able to outsurvive and outreproduce most of their competitors. As we might expect, these self-encapsulated RNA survival machines may have potentially given rise to the first cells. Sometime during the course of evolutionary history, RNA, which is single-stranded, may have experienced a copying error, whereupon it gave rise to a double-stranded copy of itself, ultimately leading to the evolution of DNA. Ultimately, these DNA survival machines would have given rise to the first cells. So therefore, we can think of cells as nothing more, at the most fundamental level, as a survival machine whereby genes facilitate their passage from one generation to another. And in the most extreme sense, we can take a look at any living organism, which of course is composed of cells, as nothing more than a survival machine that may facilitate the passage of the genetic code from one generation to the next.